Okay, everybody, just gonna quick knock this last little part out. So this part, I kind of wanted to separate this out just to go through this a little bit, just because there's a lot of kind of words and stuff here that I want us to take a second to just look at it and observe this whole chart, because this will come into play a little bit later on, okay? So we're talking about types of glycerophospholipids and sphingolipids. Okay, so glycerophospholipids are going to be the ones that we typically think of that have that glycerol backbone. So we can see down in here, it has that glycerol backbone, and it's going to have those three different chains coming off of it. It can have the two, or it can, like we see here, it has a different head group with a phosphate on it, and that will be like our diacylglycerol activated with CDP, or we can have it have an actual three individual, and then that's our triglyceride. Okay, and this is really important to understand and to remember. Whereas our sphingolipids, let me pull up a picture of those. So sphingolipids are found in all animals, plants, and fungi, and in some prokaryotic organisms and viruses. They are composed of a sphingoid base backbone to which a fatty acid may be attached through an amide bond and a head group at the primary hydroxyl. So if we look at some of these, let me find a good example here for us. These are all examples of these. So this over here is our sphingoid base right here, and then we can have an amide bond with the nitrogen, which connects us all the way through this actual chain over here. This isn't something that he usually covers super deep, but I just want to make sure that you understand that that's what a sphingolipid is. We spend more of our time focusing on the glycerophospholipids because that's more of what we see, but those sphingolipids will come into play later on as well. Okay, so like all eukaryotes, mammals also synthesize glycerophospholipids from CDP diacylglycerol or diacylglycerol. So diacylglycerol is going to be what we're going to use to synthesize some of our other glycerophospholipids. Okay, so glycero, it has our glycine backbone, or glycerol backbone, sorry, and our phospho, it has our phosphate groups here, and then lipids, these R groups here are our lipids, and from those we can make other stuff. Okay, we can come out here, we can make CMP, we can make a glycerophospholipid, all that sort of stuff. So we have the diacylglycerol being activated with CDP. This right here is going to be our CDP. Okay. So synthesis of phosphatidylcholine, phosphatidylethanolamine. Phosphatidylethanolamine is synthesized from diacylglycerol and CDP ethanolamine. Phosphatidylcholine is synthesized from diacylglycerol and CDP choline. Ethanolamine presents in phosphatidylethanolamine. It again can be exchanged for serine to form phosphatidylserine. Dihydroxyacetone phosphate serves as the precursor to the plasma logins. So the plasma logins are a different class of these glycerophospholipids. And they're going to contain a vinyl ether and an ester bond at these specific positions. I want to make sure and they play a role as endogenous antioxidants, protecting from other plasmalogens lipid and lipoprotein particles from oxidative stress. So the plasmalogens are our antioxidants. They're going to be protecting our body from different types of stress. So it's important that we can actually produce those and have those in the body. Okay, so CDP, just continue right here, CDP diacylglycerols can serve as the precursor for many phospholipids. Sphingolipid biosynthesis requires condensation of serine and palmitoyl-CoA. Ceramide is the precursor for other sphingolipids and cerebrosides. Synthesis of cardiolipin and phosphatidylinositol, and synthesis of plasmalogen. So we know what these guys do, what the plasmalogens do. Let's take a second. The cardiolipin. So the cardiolipin, here we go. The cardiolipin plays a central role in mitochondrial metabolism by maintaining the proper architecture and morphology of the mitochondrial membranes and by regulating the activity of a variety of proteins and enzymes involved in mitochondrial function. Let's go ahead and put that in here. And then we also have our phosphatidylinositol. So in plants, as in animals, they have essential biological functions. They're exerting their regulatory effects by acting as ligands that bind to protein targets via specific lipid binding domains, and so alter the location of proteins and their enzymatic activity. So let's put that in there as well. And this is where, like, as we continue down this journey of school, just taking the second to look up those definitions really quick will help you in the long run understand really what it is that we're talking about and kind of what we're actually focusing on. Okay, so now here, this chart is really cool because it kind of gives us the overlapping ideas and kind of the organization of how all of this stuff is related. We have our glycerolipids, so our glycero having the glycerol base. We have our phospholipids, so containing those phosphates, and also our sphingolipids, okay? And we can see how each of these are actually connected. So our glycerol lipids, we have our triacylglycerols, aka triglycerides. Those are for our adipose stores and our blood lipoproteins, and this is that glycerol backbone with our 3 fatty acids, or we can see those written typically as Rs, right? R1, R2, and R3. Then we have glycerophospholipids. These are some of the common examples. You'll notice that pretty much all of them, so remember cardiolipin, because that's been mentioned a couple of times, all of these will say phos. See, they have the phos base in them, so you know that they're for sure phospholipids. And then you just want to look through the rest of it. Phosphatidyl, the tidyl part right here, phosphatidyl, that is that beginning base. Every single one of those you'll be able to recognize is a glycerophospholipid. And that is a fatty acid with a phosphate group and a head group. We have our other phospholipids, our ether glycerolipids, which has the glycero-ether bond, a singular fatty acid, and then its head group. We have sphingophospholipids, such as the sphingomyelin, which we'll talk about more later. And you can see that the sphingosine base with a fatty acid and the phosphate in the head group. And then we also have the glycolipids seen here, cerebrosides, sulfatides, globosides, and gangliosides. And this is our sphingosine base with a fatty acid and glucose, and we have a carbohydrate bound there. If you have a little bit of time, I'd recommend, especially these glycolipids, as well as these ether right here, like the platelet activating factors, I would take a minute and look some of these up, just to get a better idea of kind of what they look like and what their function is, if you have the time. It's not super important right now, but if you have the time right now, it's good to do so to get a little bit of a better idea of what some of those look like and maybe how we can apply that later on. So remember the sites of bond cleavage by different phospholipases. So this is going to be our first position, our primary, our secondary, and our tertiary bonding sites. Phospholipase A1 is our primary or our first. Phospholipase A2 does the second one, and then our phospholipase C is going to break off that phosphate group there at the bottom, and our phospholipase D will act on removing the head group. Okay, take a minute to remember those. Write those down off to the side. It'd be good to write them down here. So the A1, the breaks off the first R fatty acid. A2 breaks off the second fatty acid. Our phospholipase C removes the phosphate group, and D removes the head group. Just to keep those straight. And that is all of chapter 2. I'm sure you can see that with Biochem 2, there's a little bit more dense material when it comes to all of this, so just try and keep up. Spend some time going back through this. Try 2 is definitely a doozy of a trimester. I wish you all the best of luck. Study hard, work hard, and if you have any questions, please comment down below, or you can always send me a message directly, and I can help you the best that I can. Thank you, and have a great day.